Okay, class. So today in part two, we're going to sort of look at how these various intercellular compartments sort of formed and evolved. Then we're going to look at a sort of a general overview of the secretory pathway, and then we'll cover some real specifics on nuclear transport. And so plastids, which exist in plants, are actually sort of a great model to understand sort of membrane invagination. Okay, so in plants you'll have this thing called a proplastid, and then it actually can evolve into different kinds of plastids, either a plastid for storing fats and oils or starch. You can get a chromoplast, which basically will store pigments. You see these in flowers and things like that. And then you can have the chloroplast, which is actually used to harvest light energy, which is why leaves on plants are oftentimes green. And so if you sort of look at how the chloroplast forms, what you'll have is this proplastid where you have an outer membrane and an inner membrane. And then you get these sort of invaginations of these thylakoid membranes and thylakoid vesicles, and eventually they're going to make this extra space here in the chloroplast, which is going to, be where all of the photosynthesis occurs in a chloroplast. Just as a note, I cover a little bit about plants here, but you guys don't necessarily need to memorize this. When we get into basically the electron transport chain, I'll also go over photosynthesis, but like I said, this is a class mainly devoted to mammalian cell biology, and so I don't really require you guys to memorize anything having to do with plants. But once again, this is a great model for sort of understanding how these different compartments in a cell sort of evolved, right? Because you can sort of actually watch this happen in plant cells, these formations of these plastids from proplastids. We've covered this before, and it had to do with the evolution of the nucleus. So if you have an ancient prokaryotic cell that didn't have a nucleus, but it did have some DNA, and then the DNA actually, there was an invagination, and the sort of nuclear envelope came and sort of surrounded the DNA and created a nucleus. And then what you end up having here is an inner nuclear membrane, an outer nuclear membrane, a nuclear pore, which contains the nuclear pore complex. And this turns out that it's actually contiguous with the endoplasmic reticulum, both the smooth ER as well as the rough ER. And that, invagination and sort of creating a nuclear envelope around all of the DNA converted this ancient prokaryotic cell into an ancient eukaryotic cell. And the same process is sort of believed to have occurred with mitochondria, where you had sort of an ancient anaerobic eukaryotic cell, and then all of a sudden it engulfed a bacterium, and then there was a symbiotic relationship that developed, and then this eventually evolved into what we now know today as our mitochondria. And we sort of have good evidence that this happened just because, mitochondria have actually their own DNA. Okay, so this whole thing where a bacterium got engulfed, leading to the formation of a mitochondria, and then we sort of also looked at how the nucleus was formed. And so ultimately what we're starting to see here is we have this sort of ancient eukaryotic cell here with different compartments, and then all those different compartments sort of created this transport problem that the cell had to overcome. Okay, and what we now know today is that, between these different compartments in the cell, there's actually cellular trafficking. Okay, so proteins and most secreted proteins are actually made on the rough ER, right? All translation on ribosomes occurs in the cytosol, and so anything that's made in the cytosol then needs to be transported. And what you have for a secreted protein and for membrane proteins, you have co-translational and post-translational translocation that's occurring on the rough ER, and we'll cover that in a couple of lectures. But then once the proteins are made on the rough ER, they're actually packaged into vesicles and transported to the Golgi, and then once they're in the Golgi, some other sorting mechanisms come into play where proteins can be diverted to the lysosome or to the endosome or to a secretory vesicle, and then things from the plasma membrane are actually getting sort of brought in, and they go to the endosome, and they can also then sort of go to the lysosome and other things. And we'll look at all of this sort of directional transport in the coming lectures. But here's just sort of an overview of what's happening, right? All proteins are made in the cytosol, okay? And these proteins are then sorted through various cellular mechanisms to different regions, right? You can have, in the case with the cytosol, you get this bidirectional transport between the nucleus and the cytosol through the nuclear pore complex. Okay, so proteins are made in the cytosol, messenger RNA is made in the nucleus, and that goes out to the cytosol to be translated, and proteins translated in the cytosol can go back into the nucleus. And also you can sort of think about the assembly of the ribosomes in the nucleolus. The proteins there are actually being synthesized in the cytosol, brought into the nucleus, and then the ribosomal RNA, which is being made in the nucleolus, they all get assembled into the small and large subunit of the ribosome and then get transported out. But also you have transport from the cytosol to the endoplasmic reticulum, and then you have bidirectional transport between the ER and the Golgi, and then you can see here in all of the later secretory vesicles or later secretory compartments, you also have bidirectional transport all the way out to sort of the cell exterior. You can also have transport from the cytosol to the mitochondria. Once again, all of the mitochondrial proteins are actually made in the cytosol and transported to the mitochondria, and the same thing happens with chloroplast and peroxisomes, where all of the proteins are made in the cytosol and get transported in. And so we'll actually look at this and how it all occurs. There's this little chart down here where, in the case with transport between the nucleus and the cytoplasm, it's gated transport, into the mitochondria and the endoplasmic reticulum. It's transmembrane transport, and then between the endoplasmic reticulum and all of the components of the later secretory pathway, you actually have vesicular transport, okay, where small vesicles are sort of traveling between these different compartments. Okay, and so just to give you a broad sort of overview of vesicle trafficking in a cell, what you'll have is a donor compartment. Okay, you can sort of think of this as the endoplasmic reticulum, where you have basically, membrane proteins and then luminal proteins that can then get sort of packaged into sort of a vesicle. And the vesicle actually buds off of the donor compartment, and then it's pinched off, creating a vesicle, and then this vesicle then goes on to fuse with a target compartment. Okay, and then when it fuses with the target compartment, it can release its constituents into the new membrane. Okay, and one of the things that I really want to drive home to you guys is when you're sort of thinking about this, and we'll cover this a lot more when we get to budding and fusion, especially between the ER and the Golgi. What you want to sort of keep in mind is the biochemical nature of the phospholipids that we talked about in part A of today's lecture. Okay, you have this hydrophilic head group here on the vesicle surrounding it, right? And that actually has to come together with the hydrophilic head groups here, and that has to fuse, and there's a huge energy requirement for that to occur. And the same sort of thing happens when a vesicle is actually pinched off in the whole budding process. Okay, and then there's another big question that I want you guys to all think about, and you can sort of go back to this slide to really get a good grasp of it. How do the various proteins know where to go, right? How does a cell deliver the right proteins to the endoplasmic reticulum, and how does a cell know to deliver a mitochondrial protein to the mitochondria, or a transcription factor to the nucleus, right? So that's a big question that the cell has to overcome, and it actually has kind of a simple answer. Okay, proteins get targeted to different compartments because they have different localization signals. on them. And these localization signals are oftentimes small stretches of amino acids. Okay, so this signal right here, lyse, 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 arg, lyse, is actually a signal to get into the nucleus. So that's a nuclear import signal. Okay, you also have ones that can tell something to be imported into the mitochondria or into a plastid or into the endoplasmic reticulum. And these are somewhat highly conserved. There is some variation between them. You guys aren't required to memorize any of these except this last one, which is an ER retention sequence. Okay, and that's lyse, aspartic acid, glutamic acid, and leucine, and it's abbreviated KDEL. And we'll actually look, when we're talking about retrieval from the endoplasmic reticulum, you know, if an endoplasmic reticulum protein gets somehow transported to the Golgi, it has this sequence on it and it's bound by a receptor and retrieved. And so, like I said, you don't need to memorize this right now, but as for localization sequences, this one's small enough that you guys should be able to memorize it, right? It just is KDEL. That's the amino acid sequence for it. Okay, and like I said, we'll cover that again. But it's these sort of localization sequences that dictate where a protein's going to go. And you guys can almost think of these almost as like zip codes, right? The post office knows where to deliver mail because they have address labels and zip codes, right? The zip codes tell you the town and the address tells you the street. Okay, so you can sort of think about these as biological zip codes. Okay, so now we're going to actually start to cover nuclear transport, okay? And as I mentioned just a few slides ago, it's bidirectional, right? Proteins are made in the cytosol, messenger RNA is made in the nucleus, and there's sort of this bidirectional transport. Okay, so all proteins are synthesized in the cytosol. You can think of sort of the transcription factors, histones, DNA polymerase, RNA polymerase, all the DNA repair enzymes, the general transcription factors. You know, they're all made in the cytosol and transported into the nucleus. Whereas messenger RNA and ribosomal RNA is actually made in the nucleus, and the ribosomes are actually assembled in the nucleolus, and they get transported out into the cytosol. Okay, and so I just want you to sort of really appreciate there's this bidirectional transport here, and it's a regulated process. Okay, and if we look at sort of a cross-section of a nucleus, what you have here is the nuclear envelope, which has two membranes, right? There's an inner nuclear membrane and an outer nuclear membrane. Okay, and then there's this sort of perinuclear space that goes around it. And between it, because it's an envelope and not just a lipid bilayer, you actually have these nuclear pores. Okay, and inside the nucleus you have the nuclear lamina. Okay, and we sort of saw that before when we were covering intermediate filaments. One other thing that you need to know is that the nuclear envelope is contiguous with the endoplasmic reticulum. Okay, so it just sort of, once you get past the nuclear envelope, there's still a continuation of the lipid bilayer, which then makes the ER membrane. Okay, and then you have an ER lumen here. Okay, and so one other thing that I think you guys is important to know is that the inner nuclear membrane is actually different from the outer nuclear membrane. And all of the transport in and out of the nucleus occurs through these nuclear pores. And the complex that actually facilitates all that transport is called the nuclear pore complex. Okay, so here's some great electron micrographs of the nuclear pore complex. And you can see the symmetry that exists in it. There's actually, I think, 8-fold symmetry in this thing, in the nuclear pore complex. And here's sort of a little schematic of it, right? You have these little cytosolic filaments that are coming out to help capture stuff, and then you have your ring subunits that go all around it. You have this annular subunit on the interior, sort of shaded green here, and then you have these column subunits that are sort of spread around it. And then in the nucleus itself, you have this thing called the nuclear basket. Okay, and these nuclear baskets have basically these little nuclear basket filaments that come down, and then you have the sort of the catch can down at the bottom here. And so here's basically another sort of cross section of an electron micrograph showing the nuclear pore complex and how things sort of get through it. Okay, so in total there's about 30 proteins and 8-fold rotational symmetry. So if you were to turn it, there'd be that symmetry, and then there's also 2-fold transverse symmetry in it. Okay, now one of the things that you need to know is that transport in and out of the nucleus can be either active or passive, and it partially depends on the size of the molecule. Okay, small molecules can freely diffuse in and out of the nucleus. Okay, and that'll be based on the concentration gradient on whether or not they'll go in or out, and they'll be sort of reliant on equilibrium. However, larger molecules are actually actively transported into the nucleus, right? Because they can't just go through. So small molecules can freely diffuse, like small transcription factors and stuff like that. Whereas larger molecules need to be transported in and out. So these larger molecules are going to need either a nuclear import signal to be brought in, or a nuclear export signal when they need to be brought back out. And we'll see that sometimes proteins contain both, and depending on the phosphorylation state of the protein, it will tell it to either enter the nucleus or exit the nucleus. And like I said, we sort of covered the nuclear localization sequence already. Here's a classical one, lyse, 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 arg, lyse. And you can see if you just put this signal on a random protein, it will actually enter into the nucleus. So here are basically stained nucleus, and this is the T antigen containing a nuclear import signal, and gets brought in. And if you mutate that signal, all of the protein is actually out in the cytosol. It doesn't get imported in. Okay, and in the case with nuclear localization sequences, this is a very easy one to spot. Okay, but sometimes they're a little bit more complicated than that, and oftentimes they occur as patches on a protein. So it's not always readily apparent what proteins have a nuclear localization sequence and which ones don't, just by looking at the amino acid sequence of the protein. Another thing that I think you guys might find interesting, and we covered this earlier when we were talking about gene editing and CRISPR-Cas9. So one of the real breakthroughs with gene editing in mammalian cells and the use of CRISPR-Cas9 in that process was, if you remember, the Cas9 nuclease, it's actually of bacterial origin and doesn't normally enter the nucleus. And in order to get it to work in a mammalian cell, they actually had to put a nuclear localization sequence on the Cas9 nuclease to actually get it to enter the nucleus in a mammalian cell. And that was one of the major breakthroughs in that whole process, so that it could be used for gene editing in a mammalian cell. Okay, and that's why some of this stuff's important to you guys. Okay, so here's just a great little schematic, or a great little electron micrograph of active nuclear transport. So what they did was they took these colloidal gold beads here, and they actually put a nuclear localization sequence on it, and you can see they added it to it, and you have your nuclear sort of envelope here, and then the nuclear pore complexes, and they can actually watch these beads sort of enter the nuclear pore complex and then be eventually transported to the other side of it and brought into the nucleus. Okay, so I'm sure you guys are all dying to know how nuclear import works. Well, it's mediated by these proteins called nuclear import receptors. Okay, and there's a whole class of these different proteins. Okay, so here they're sort of just all drawn as S's, and the import receptors are all going to sort of recognize different nuclear localization sequences, and in some instances you can actually have adapter proteins that help bring cargo proteins in, and the adapter proteins will actually interact with the nuclear import receptors. Okay, and these nuclear import receptors, they basically all sort of associate with the nuclear pore complex, and that have these sort of FG repeats, and that helps to bring cargo in, okay, into the nucleus. And it's a very regulated event, and how this whole thing occurs. And part of it is based on this small G protein called RAN, okay? And what's really important in all of this is that you have the RAN gap on one side of this nuclear envelope and the RAN GEF on the other side. Okay, so the RAN gap is in the cytosol and the RAN GEF is in the nucleus. And this is basically causing, the RAN GEF is going to cause the exchange of GDP for GTP, and then when the RAN GTP gets out, it actually gets hydrolyzed into the GDP bound form and inorganic phosphate, and then it can be brought back in again where the RAN GEF will exchange the GDP for GTP, and then you've got your RAN GDP. Okay, so all of nuclear import and export is dependent on this small G protein called RAN, as well as the nuclear import and export receptors. Okay, and this is really sort of a key point that I want you guys to all know, because here's basically how it works. Okay, in the case with nuclear import, okay, what you're going to have is your nuclear import receptor. Okay, that's going to bind some cargo, and that's going to basically bring it into the nucleus. And then you'll have an abundance of RAN GTP in the nucleus because that's where the RAN GEF is. And the RAN GEF will basically bind to the nuclear import receptor and release the cargo that was there into the nucleus. And then the nuclear import receptor with the RAN GTP actually exits the nucleus, where it meets the RAN gap, and that causes hydrolysis of the GTP to GDP, releasing inorganic phosphate, and then that's all released from the nuclear import receptor, so that the nuclear import receptor can undergo a second round of bringing in new cargo, and then the RAN GTP binds to it, right? Now, in the case with export, it's a little bit different, okay? And so what ends up happening is you'll have your protein with a nuclear export signal that'll bind to a nuclear export receptor, and that will also then associate with the RAN GTP, and that's all brought out of the nucleus, where the RAN gap then hydrolyzes the GTP to GDP form, and that releases everything. The cargo to be delivered to the cytosol is released, as well as the RAN GDP bound form, and the nuclear export receptor can then be brought back into the nucleus for its next protein to be brought out. Okay, so once again, it's very important that you guys sort of appreciate the role of the RAN gap and the RAN GEF and the RAN and the small G protein RAN in this whole process, and how it interacts with the nuclear import and export receptors to bring things in and out of the nucleus. Here's basically the structure of a nuclear import receptor. You have it here, and here you have the RAN GTP. So this would actually be in the nucleus at this point in time, and then you have your cargo binding. So this will be brought out. The RAN GTP will be hydrolyzed to RAN GDP, and the RAN GDP will be released, and then the import receptor can bind cargo and then be brought back in. Okay. And the cargo loading and unloading actually has to do with that little switch loop, just like with other things. So it can bind a cargo, and then when the cargo is unloaded, the RAN is replaced, the RAN GTP is replaced, and the cargo is released. And then once this gets to the cytosol, the RAN GTP will be hydrolyzed to RAN GDP, releasing it from the import receptor, and then a new cargo protein with a nuclear import signal can bind and be brought back into the cell. Okay, so this sounds all pretty basic and straightforward. However, it's actually regulated in a lot of ways. So certain proteins will only get incorporated into certain into the nuclei of certain cells. Okay, and so you can sort of see that here in sort of a Drosophila embryo, where this protein that's sort of brownish is being imported into these nuclei, but is not being imported into these sort of outer nuclei. So once again, there's sort of regulated nuclear import. Certain cells will have certain receptors that will allow certain proteins to be brought into only certain nuclei. Okay. And as I mentioned before too, oftentimes the phosphorylation state of a different cargo molecule can determine whether or not a protein is in the cytosol or the nucleus. And this happens a lot, especially with cell signaling and whether or not a transcription factor is going to be in the nucleus or in the cytosol. Okay, and so you have this protein NFAT, and in the cytosol, when it gets exported from the nucleus, it's phosphorylated. It'll bind to this calcineurin protein, which is a phosphatase, and it'll remove the phosphates. And the removal of the phosphates sort of exposes this nuclear import signal, and then that will actually get brought in by an import receptor. And then this protein can then go on to activate transcription. And during the whole process of activating transcription, it will actually get phosphorylated, and the protein becomes phosphorylated. And once it's phosphorylated, that actually causes the protein to sort of change, and the nuclear import signal is sort of buried, and you get an exposed nuclear export signal, which is then bound to an export receptor and brought out into the cytosol. And then once again, it's going to encounter the protein phosphatase, which removes the phosphate. And so certain transcription factors can actually cycle in and out of the nucleus depending on the needs of the cell. Okay, and you can sort of think of the phosphorylation as masking the nuclear localization sequence, and that can influence the import and export. Okay, finally, I just want to re-emphasize the role of nuclear lamins and nuclear lamina. It's an intermediate filament. We've sort of already covered it in different parts, but here it sort of creates this sort of mesh-like structure on the outside of the sort of the internuclear membrane, and it's bound there, and oftentimes the nuclear lamina will be sort of where the telomeric heterochromatin and other heterochromatin in the cell resides. And when the cell undergoes cell division, this nuclear lamina is actually broken down. Okay, and so what you have is an interphase nucleus, then you have your nuclear lamins that are on sort of creating this sort of associated with the nuclear envelope, the inner membrane of the nuclear envelope, and then you'll have your DNA, and then when the cell starts to divide and you get into prophase, and we just covered the secretory pathway, the lamins become phosphorylated and the nuclear envelope starts to break down, the chromosomes condense, and then once you sort of get through the metaphase to anaphase transition and you enter into early telophase, the chromosomes start to decondense and the nuclear envelope starts to reform, and eventually you'll end up with two individual nuclei with nuclear pore complexes in them. Okay, and then left back with your interphase nucleus. Okay, so that's pretty much all that I had for today. So next Tuesday, we're going to cover more on membrane proteins, and then translocation in the ER. So I encourage you guys to do the reading ahead of time. And also a reminder, this coming Thursday is going to be your second exam, and that'll be on March 25th. And I will talk to you guys all later. Have a great day. Bye-bye.